Hey there, welcome back. So the next tool we're going to look at is gel electrophoresis. So what DNA gel electrophoresis does is it runs DNA within a gel from one end of a gel to the other end using electric shocks, basically. So one end of the gel is put into an electrolyte solution, and one end is given negative charged electrodes. The other side is positively charged. DNA is going to run from the negative charged electrodes towards the positive charged electrodes. And it moves associated with its size. So this is a gel, and gels have small pores. And so small sections of DNA are going to run faster than larger sections of DNA. And in the end, you're going to get a series of lines that are associated with different sizes, and then you can determine you can look at fingerprints of a suspect, or you might look at if there's a positive or a negative mutation within some DNA. You might see if, the paternity of an infant. All of those things can be determined by using gel electrophoresis. The last tool that I'll talk about is the CRISPR system. So CRISPR systems, which we talked about also in Chapter 8, which are used as part of the immune system of bacteria, can also be used by us in biotechnology. So in CRISPR systems, bacteria have DNA cutting enzymes known as Cas enzymes, and these enzymes cut viral DNA up, and then they can store them. What we use is a modified form of Cas9, and we use our own RNA that we control. The RNA, single-stranded RNA, goes in and binds to a specific sequence of DNA, and then the Cas9 enzyme cuts the DNA at the specific site. So we control where we're cutting the DNA. And this allows us to manipulate the cell's normal DNA, and we can then modify the DNA however we want. I'm trying to move to the next slide. Hello. There we go. All right. We also can use a modified form of Cas9 nuclease that doesn't digest the DNA. So because it doesn't digest the DNA, it is called a dead or D-Cas9. All right. But it still uses the RNA to guide to a specific region of DNA. And then that RNA can actually block transcription. It can also be used to be carried into a cell. We can add onto that RNA a sequence, an activator enzyme that could activate protein synthesis. We could produce a D-Cas9 that carries a fluorescence marker. So when it goes in, it fluoresces, and then we can see where that gene is located within a cell. So there's a lot of purpose for these D-Cas9s. So we have a lot of applications of genetic engineering. Some I wrote over here. You can see a whole list over here. Protein production, big one. DNA production, research, and cloning are some of the major things. When I think of protein production, we produce proteins. If you look here, all of these can be used to treat different disorders. Okay, genetic engineering for vaccines, more proteins, DNA production so we can study things. And then we have transgenic plants, transgenic animals, all down here, and of course cloning organisms and cloning DNA. So how do we do this? I love this figure. This shows bacteria over here that contain certain chromosomes. There's a plasmid. Plasmids are all often known as vectors. So I'm going to put that over here. And a vector is something that carries material. To a cell. All right. So we talk about vectors, like I've talked about vectors with, I don't know if I have in this class yet, but I often talk about vectors as insects that carry disease from one organism to another, or from some material to another set of material, and then that can transmit the disease. In this case, the vector is carrying genetic information, and it is delivering that genetic information to another cell. So if we take the plasmid out of a bacterial cell, and we take out some DNA that we care about, we cut both with a restriction enzyme. We then allow the gene that we care about and the plasmid to anneal together. We then add the plasmids into a solution of bacteria and allow the bacteria, competent bacteria, so let's call it competent, to take in the vectors, the plasmid with the gene that we care about, right? We then have produced a recombinant cell. That cell can then be used to produce special characteristics within the bacteria. In this case, we have produced bacteria that can clean up oil spills, right? Or toxic waste. So recombinant. Over here, we can take the bacteria that are now recombinant and we can harvest proteins produced. And these proteins might be used in making clothes or in treating diseases. All right, I'm going to stop here on this video and we'll get into biotech a little bit further in our next video. Bye.